Good afternoon, ladies and gentlemen. Welcome to Bio with Friends. Today we're here with American scientist Thomas Hunt Morgan. How are you on this beautiful afternoon, Thomas?
I am pleasantly well, thank you for asking.
Well, that's great to know. Today we're going to be talking about gene linkage. Gene linkage was first described by William Bateson and R.C. Punnett. Punnett and Bateson, like Mendel, studied dihybrid crosses of pea plants. The results suggested that some genes were linked together. Professor Morgan, could you please explain to us how can genes be linked together and follow Mendel's law of assortment?
Well, we started working with fruit flies, also known as Drosophila melanogaster, and we found the answer. At first, I was just looking for an organism to use in genetic research. I found fruit flies very useful because I could quickly and cheaply grow new generations of flies. I observed among fruit flies easily identifiable variations in eye color, body color, and wing shape. Knowing these variations, I and my students set up experiments similar to Mendel's dihybrid crosses. I chose one type of fly with traits associated with the wild type, or most common phenotype. I crossed the wild type flies with mutant flies, or flies with a different, less common phenotype. Do you have any questions?
What were your results and how did they differ from Bateson and Punnett?
Well, my results, like those of Bateson and Punnett, did not always follow the 9:3:1 ratio predicted by Mendel. But the results did differ in a noticeable pattern. Some traits appeared to be inherited together. I called these traits linked traits, and they appeared to fall into 4 groups. As it turns out, fruit flies have 4 pairs of chromosomes. Each of the 4 groups of linked traits identified by myself matches one of the chromosome pairs. I concluded that linked genes were on the same chromosome. The chromosomes, not the genes, assort independently during meiosis. Because the linked genes were not inherited together every time, I also concluded that chromosomes must exchange homologous genes during meiosis. Well, it's been a pleasure being here in this interview. Thank you very much, Dr. Iqbal.
Well, it was great having you, Thomas. I wish you good luck in your future research.
Thank you, I do appreciate it. Have a good day, everyone. Stay safe.
Hungry Bunny, I'm so hungry. Hungry Bunny, I'm so hungry. Hungry Bunny, come and have a super bunny right now.
Beef grilled burger, cheese and crispy lettuce, onion, fresh tomato,
All put together in a sesame seed bun. Super bunny, fish sandwich. Hungry Bunny, chicken too. Hungry Bunny, come and have a Hungry Bunny right now. Hungry Bunny, come and have a Hungry Bunny right now.
Hello and welcome back to Bio with Friends. Sorry Dr. Iqbal couldn't be here today. He was killed in a freak horse riding accident. Anyways, my name is Professor Noah, and we are here with Alfred Sturdivant. So Alfred, how are you today?
I'm doing pretty well. How are you?
I'm doing great, thank you for asking. So Alfred, please tell us, what have you hypothesized in the past few years?
Well, I hypothesized that the frequency of crossovers during meiosis was related to the distance between genes. This meant that the closer together 2 genes were, the more likely they were to stay together when crossovers took place. I identified the linked traits in fruit flies: body color, eye color, and wing size, and then crossed the fruit flies. I recorded the percentage of times that the phenotypes didn't appear together in the offspring. This percentage represented the frequency of crossovers between chromosomes. From the crossover frequencies, I made linkage maps. The linkage maps were maps of the relative locations of genes on a chromosome.
Well, thank you, Alfred. That has been quite enlightening. Do you have any further comments or questions to add?
Well, I have a little joke for you, Mr. Perry.
How pleasant.
If there are 5 fruit flies in the kitchen, how do you know which one's a football player?
I don't know.
The one that's in the sugar bowl.